for having me. So Spectris is a medical device. It's an integrated headset that delivers patterned light and sound to evoke a brain response, an electrical brain response that is in a very specific frequency of brain activity called the gamma frequency. And the device in the future will be physician prescribed. It will be used in the home by patients after the device is delivered to the home and the settings are adjusted. And then patients with Alzheimer's disease will use it for 1 hour a day in the comfort of their home. So I'll start with how it works. The preclinical and clinical clues that we have suggest that the gamma oscillations that are generated spread through quite a wide range of brain structures, including deeper brain structures. And as a consequence of that, it activates a type of neuron in the brain, which is an inhibitory neuron called a parvalbumin neuron. And these neurons are the pacemakers of the brain, and they control the oscillations in different regions and between regions. And as a consequence of activating that group of inhibitory neurons, there's a release of synaptic and myelin proteins in the various structures in the brain, including neurons and then also the glial supporting cells. There's also a reduction in neuroinflammation. And as a consequence of those biologic changes, we then see improvements or a reduction in loss of daily function, also cognition. And very importantly, we see very prominent preservation of brain structures. We can measure the whole brain or parts of the brain. And the part of the brain where we see a very interesting signal is in the white matter. So we see preservation of white matter content in all brain regions. It's most noticeable in the entorhinal cortex, which is the part of the brain in the temporal lobe that's very much involved in Alzheimer's. And most recently, we looked at a structure in the brain called the corpus callosum, which is that interhemispheric highway that has about 250 million myelinated axons. And we show a remarkable preservation after 3 months and 6 months of corpus callosum area, which is a new finding in Alzheimer's disease. We're very excited about learning more about that. We're in our 5th clinical trial right now. The technology came out of MIT, out of the labs of Li Wei-Tsai and Ed Boyden. And the first studies that were done were to prove the biology. In other words, to show in humans that the same type of changes that were seen in animals could be seen in humans with Alzheimer's disease. And the main things that were looked at were connections, electrical connections in the brain, so-called network connectivity, and also brain structure, so looking at the shape and size of different parts of the brain. And also clinical outcomes such as verbal memory. And in addition, one of the first studies looked at the changes in the spinal fluid protein levels of different proteins that reflect important functions that could be modified by the disease. So specifically, we looked at proteins that are involved in synapses and myelin, the lining around axons. And we showed very interesting changes there that told us that we were probably on the right path of connecting the changes in structure and function to those core biological activities that are induced by the gamma oscillations that we can produce in the brain. So after the proof of biology studies, we undertook a proof of concept study, which was a randomized controlled trial called a phase 2 trial called Overture, where we were able to randomize in 2-to-1 ratio onto active settings or sham settings of the device. And we're able to show after 6 months that there was a remarkable preservation of function in terms of activities of daily living and cognition. And in addition, at the same time, we measured brain structures. So we looked at MRI brain structures to show that we could slow the rate of atrophy. And the two results were very concordant. In other words, we showed a very similar pattern in terms of daily function and cognition and preservation of brain structure. In addition, we didn't see any serious treatment limiting adverse events, which was a very important finding. We didn't see ARIA, which is an adverse event related to a certain class of medications. And we saw about 80% device adherence over 6 months. That was followed by a year of open label extension where people had the option of continuing on treatment or switching to active treatment. And we showed that we had durability of treatment out through 18 months. And also those who switched from sham treatment to active treatment obtained new benefits that they didn't have before. So the rate of progression of functional loss and brain atrophy were then very similar to those who were treated on the active device from the start. So coming into CTAD, we obviously had a lot of excitement, and we're really privileged and excited to share new data here today.